Good evening, folks. This is Natalie Mantegna reporting for 6ABC News. Today is the day that hundreds of years ago, Robert Hooke in 1665 discovered the cell using a microscope. And to commemorate his special discovery, we have a special guest today. Say hello to the cell, the plant cell. As a refresher, today we have a plant cell, which is a eukaryotic cell, which has a nucleus and many cell organelles that perform specific jobs. On the other hand, there are prokaryotic cells which don't have nucleuses or as many organelles and include the bacteria and archaea and are adapted to live anywhere. Using modern-day microscopes, we can zoom in to see the different organelles in different types of cells, which tell us how they work and how they function. You can tell this cell is a plant cell as opposed to an animal cell because it has a cell wall, chloroplasts, and a large central vacuole. The cell wall protects and supports the cell, so you could think of it as a backbone or a skeletal system. The chloroplasts use solar energy to make sugars for photosynthesis, which is how the plant survives and gets energy. The large central vacuole is larger in plants than in animals because plant vacuoles are typically filled with fluid to give the cell added support, such as water, sugars, salts, pigments, and toxins. The cytoplasm is the general area between the nucleus and the plasma membrane, so all of these organelles. If you don't recall, organelles are tiny specialized structures performing specific cellular functions. Take this vacuole for an example. The plasma membrane, not to be confused with the cell wall, surrounds all cells and regulates what materials enter and leave the cell. The endoplasmic reticulum, otherwise known as the ER, is a system of membranous channels and saccules or flattened vesicles. The rough ER has ribosomes and makes proteins that will be used outside of the cell, whereas the smooth ER lacks ribosomes, and it's the site of synthesis of complex carbohydrates and lipids, including phospholipids, packaging of proteins into vesicles, and transported to the Golgi apparatus, which is here. The ribosomes floating around the cytoplasm, otherwise called as the fluid, which is the cytosol, proteins are made here are used by cells at structures such as mitochondria, which are here, and chloroplasts. And ribosomes on the ER are sent out from the cell to become part of the cell membrane, which is here. The Golgi apparatus, here, here, and here, receives vesicles containing proteins and lipids from the smooth ER, packages, processes, and distributes them within the cell, and it's also involved in secretion and the formation of lysosomes. Lysosomes are involved in intracellular digestion. When cells bring in macromolecules, vesicles fuse to lysosomes and contents are digested. Think of the lysosomes as the stomach. The mitochondria here, here, and here breaks down sugars to produce ATP, and it's involved in cellular respiration, which coincides with photosynthesis, which was talked about earlier. It's found in plant and animal cells, and it's surrounded by a double membrane. Photosynthesis takes solar energy and carbon dioxide and water and turns it into carbohydrates and oxygen, and cellular respiration takes in carbohydrates and oxygen to turn it into carbon dioxide, water, and energy. The cytoskeleton maintains cell shape and causes cell and its organelles to move, and that contains microfilaments and microtubules. The microfilaments mainly help the cell take shape and enable the entire cell or parts of the cell to move, whereas the microtubules form rigid skeleton for the cell, assist in moving substances within the cell. And the centrosome is the microtubule organizing center. The cilia and flagella are involved in cell movement. The cilia has small, numerous hairs, and the flagella, oppositely, has large, single hair. The 9+2 pattern of microtubules is what makes that up. Here we have the most important part, the nucleus. Think of it as the brain of the cell. It stores genetic information and controls metabolic activities, and the nucleus contains chromatin, nucleoplasm, the nucleolus, the nuclear envelope, and the nuclear pores, which are shown on the outside here. The chromatin that resides inside the nucleus contains DNA and protein and will coil up to form chromosomes before cell division. The nucleolus inside the nucleus makes ribosomal RNA, which will leave the nucleus to form ribosomal subunits in cytoplasm to make proteins. The nuclear pores are openings in nuclear membrane to allow proteins into the nucleus and RNA out of the nucleus. The nuclear envelope is simply the double membrane around the nucleus, and the nucleoplasm is the semi-fluid material inside of the nucleus. And that's it for the plant cell. Oh, it looks like we have another guest for tonight. Who is this? Introduce yourself.
Animal cell.
Nice to meet you, animal cell. How are you doing tonight?
Good.
Great. Here we have the animal cell. This is the cell that we have in our bodies. The difference between the animal and plant cell is that the animal cell has no cell wall. It has centrioles. It has more smaller vacuoles around. Has more lysosomes. The organelle of the centriole is involved in cell division. The animal cell has more small vacuoles because it does not need to have added support since it is a heterotroph, meaning it gets its food from eating other organisms as opposed to plants that are autotrophs that make their own food. There are more lysosomes in the animal cell than the plant cell because lysosomes are involved in intracellular digestion and animals are heterotrophs, meaning they have to digest their own food. And think of lysosomes as the stomach. Here's the rough ER, the smooth ER, the microtubules, the microfilaments, mitochondria, the vacuoles, microtubules, microfilaments, more ER. Here is the nucleus, the chromatin, the nucleolus, the nuclear pores, nuclear membrane, double membrane there. Here is the Golgi apparatus, mitochondria here, the centriole, vacuole here, mitochondria, lysosomes, vacuole there, and around here is the plasma membrane. And with that, folks, that summarizes up our evening contribution to Robert Hooke, who discovered the cell first in 1665. I would like to thank, on behalf of myself and 6ABC, I would like to thank our special guest, the animal. Thank you, sir, for being on the show.
Thanks for having me. I got a split.
Boy, that guy's funny. Anyway, good evening, folks. I hope you have a great night. This is Natalie and Mantegna signing off from 6ABC.